Good afternoon. Welcome to this week's lecture, which is the G. Burroughs Miter Lecture, in honor of the NIH First Director of Laboratories and Clinics, which I'm told was the predecessor to my job as Deputy for Intramural Research. Before I introduce today's amazing speaker, let me make a couple of announcements. We will not have a WALS next week. The next WALS will be on March 11th. It will be the Margaret Pittman Lecture given by Jennifer Doudna, and it will be at Natcher Amphitheater, not Missouri. The reason for this is, first of all, we expect a big audience, but mostly because there's an RNA conference, and she's going to be also part of that conference. It will be at 3, March 11th, Pittman Lecture. So the lecture in honor of G. Burroughs Miter, who was known around NIH as Bo Miter, was to recognize our most outstanding intramural scientists, and we certainly have done that today. Our speaker is Crystal Mackall, the chief of the pediatric oncology branch in NCI. She has an undergraduate degree from the University of Akron in Akron, Ohio, and an MD from Northeastern Ohio University's College of Medicine, with training at the Children's Hospital there in internal medicine and pediatrics. I guess she decided to leave the Midwest and come east, and did a fellowship in pediatric oncology with Ron Gress in the pediatric oncology branch, and then worked subsequently in experimental immunology, and returned to pediatric oncology branch, where she became first a tenure track investigator, and about a dozen years ago was tenured as a senior investigator at the NIH. Her research has focused on the interactions between the immune system and cancer. Initially, I think the interest was more in the way in which cancer affected the immune system, particularly how chemotherapy affected the immune system. It was clear that patients, particularly pediatric patients who were getting a lot of chemotherapy for leukemia, had really profound changes in their immune system. And so she began to study the homeostasis of T cells in patients with cancer, and actually developed an important immunorestorative interleukin-7 in the treatment of children with pediatric cancer. More recently, I think her work has morphed into the use of cells to treat cancer, particularly adoptive cell therapy of cancer. You're probably all aware, because this has gotten a lot of press and really deserves a lot of press, is an amazing new way of treating cancer. That her particular work is focused on the use of T cells expressing the CD19 chimeric antigen receptor to treat pediatric B-cell acute lymphocytic leukemia. And she's published what I would just call amazing and dramatic results, and I suspect you'll hear more about that work today. Her leadership in this field of cell-based therapy is recognized in a variety of ways, including very recently being asked to head a Stand Up to Cancer project to develop cell-based therapies for cancer. She's an elected member of the American Society for Clinical Investigation, and has received many NIH awards, including a Distinguished Clinical Teacher Award, an NCI Mentor of Merit Award, and several NCI and NIH Directors Award for her scientific accomplishments. In addition, she's just a wonderful NIH citizen. Crystal hardly ever says no when there's an important job to be done at the NIH. And for many years, she's been an informal advisor to me on matters about clinical research. So let's welcome Crystal Mackall. Her talk today is entitled "The Immune System in Childhood Cancer, Mobilizing the Troops."
Thank you, Michael. Thank you, Michael, for the kind introduction, for your support through the years, and to the NIH leadership for providing me with this opportunity to speak as part of this distinguished lecture series. In preparing this talk, I wanted to find out more about G. Burroughs Miter, and was really happy to hear that he was described as the first director of laboratories and clinics, because, you know, the ring of that was sort of right up my alley. Because as you will see, as I kind of go through my career here a bit, in this talk, my career here has really been a celebration of the NIH's both laboratories and clinics. And I hope that I can demonstrate to you that work such as this cannot be done without the deep scientific expertise that populates the bench laboratories around this campus. And it cannot be done without the world-class clinical research program that is embodied in the NIH Clinical Center. This work is really a synergy between these two amazing resources that I've had the privilege to utilize over the last 2.5 decades. So the reason I'm here actually today started on a day in 1985 when I was actually an intern at this hospital in Akron, Ohio, had already been interested in oncology and in immunology, and I read this article. It was a special report in the New England Journal of Medicine using IL-2 activated immune cells, administering them to patients with cancer and giving high-dose IL-2. And I'd really never heard of anything like this before, but I was enamored of the notion of using the immune system to treat cancer, and of course preliminary, but there was some interesting results here, one complete response. And I wanted to know, who in the world was doing this, and it was this place called, the National Cancer in Bethesda, Maryland. I thought, okay, that's the place I want to go. So this was 1985, and around 1989, I had finished my residency and was really very happy to have the opportunity to enroll in the fellowship program in pediatric oncology at the NCI pediatric branch. And at the time, the branch was being led by a man named Phil Pizzo, who really was quite a mentor for me. Not a scientific mentor per se. Phil was mostly a clinical researcher, but a very charismatic leader, and demonstrated to me how important it is when leading scientific groups towards goals that aren't always difficult, how important it is to have this kind of charismatic leader in your midst. And probably the message that Phil left me with, which still resonates with me today, is that as a pediatric physician scientist, it's not just an option to try to create new therapies for our children, but it's an ethical imperative. And that may seem obvious to people in this room, but I will tell you that there are people, and there are people even today, who have trouble thinking about doing investigative studies for children with dread disease. It's sometimes easier to say, well, they're a vulnerable population, and maybe you'll be pushing an ethical boundary. But Phil really taught me, and I agree, that it's our ethical imperative to try to take the advances that are being made throughout medicine and bring them to our children. Now, he was also interested in HIV infection, and so we had a lot of kids with HIV on the ward when I first came, and I also got interested scientifically, as Michael alluded to, in this problem of T-cell lymphopenia. And at the time, we really had very little understanding of how the immune system, how the body is able to respond to an insult that depletes T cells. And so I needed to find a scientific mentor, and I was lucky enough, and I mean this absolutely lucky enough, to find Ron Gress, who was leading the transplantation immunology section in the experimental immunology branch at that time. And of course, interested in immunotherapy of cancer, and at that time, certainly bone marrow transplant was, and still was, the first effective immunotherapy for cancer. And so I went to work with Ron, and Ron taught me so much, not only about science, but the culture of science. He was also a physician who understood how to make that link between the bench and the bedside. And we got really interested in this problem of T-cell depletion. This was the clinical vignette that I remember. I'm sure Ron remembers it as well. This was a 15-year-old with high-grade brain tumor treated in the pediatric branch. And at that time, these myeloid growth factors had just come out. And all of the world of oncology was very excited about this. We thought that if we could only give chemotherapy more frequently and at higher dose, and at the current time it had been limited by neutropenia, if we could give more chemotherapy, we could cure more patients with cancer. And so using GM-CSF, we started to be able to give really high doses of cyclophosphamide every 2 weeks. It was remarkable that we got away with it in terms of the neutropenia wasn't too bad. There weren't a lot of fevers. And this one patient went home to his local hospital in between cycles. He wasn't feeling sick. He needed a platelet transfusion. Received the platelets at a local hospital that weren't irradiated and came back to us with a devastating complication, transfusion-associated graft-versus-host disease, for which he ultimately died. Now, this vignette, I think to a physician, it teaches you that, the importance of irradiating blood products, et cetera. But to the scientist in us, it was really a red flag that something is happening with these patients that we don't understand. We had no idea how their immune system could be so weakened by the cyclophosphamide and why that would be the case when their neutrophils were recovering so well that they couldn't even reject the few T cells contained in a platelet transfusion. And this was, of course, associated with other complications that we were observing. And so we began a bench-to-bedside-to-bench project focused on the problem of impaired immune reconstitution in humans. And this is just a little bit of raw data from some of our early publications. And again, you just see how well, with each cycle of chemotherapy, most of the cell populations recover very quickly. But look here in red. The lymphocytes just get worse and worse. And there really wasn't a good pathway to make these lymphocytes come back. But we had no idea what the rate-limiting factor was. And so I went into the lab using mouse models and began to dissect how the immune system the T-cell immune system in particular, can regenerate itself after it's been depleted by disease or therapy. And, some of this, there was a sort of a general understanding of it, but it really was not very well fleshed out. And, of course, there was the recapitulation of ontogeny, where hematopoietic stem cells go through the thymus and regenerate a nice, diverse repertoire. But there was this other pathway that wasn't well understood called homeostatic peripheral expansion, which was not as good at regenerating repertoire diversity, but could become very dominant. And then there was this kind of vestigial-type pathway, which wasn't very effective, but people like to talk about. And we ended up demonstrating that these were the two major pathways, and that you could distinguish progeny of these pathways using the phenotype on T cells by flow cytometry. And with that tool in our pocket, we then went back to the clinic and we said, all right, we're going to figure out what are our patients doing? Why aren't they able to regenerate? What pathway are they using? And using patients that were treated in the pediatric branch, in this case for lymphoma, we were able to identify the profound CD4 depletion that is so emblematic of cytotoxic chemotherapy. And we noted that while the patients started with both naive and memory cells, in this case, you end up losing all of your naive populations, and it's only upon the T-cell recovery in this 3-year-old that we saw recovery of naive cells. And so we predicted that this represented a biomarker for thymic activity. And when we went ahead and looked at older populations, even teenagers, we never did see, or for a very long time, did not see recovery of T cells or recovery of naive cells. And so we identified that age was the single most important factor that impacted the ability of the human to be able to regenerate their immune system. And remarkably, relatively early in life, humans lose this capacity to regenerate all via the thymic-dependent pathway. And so most of our patients that we were treating, we were really subjecting them to the reliance on these other pathways. And we were able to image the thymus and show that the young patients had the big thymus and the older patients didn't. So it all really made for a very nice story. And at that point, then, we began to turn our focus towards the problem of, well, it wasn't going to be easy to turn the thymus on, and actually Ron has worked on that since that time, but I really turned my focus to this homeostatic peripheral expansion pathway, the other pathway by which we can make T cells. And as imperfect as it is, it's turned out to be something that is an important tool now in the treatment of cancer with immunotherapy. And to that end, I was lucky enough to recruit a young investigator, Terry Fry, into my laboratory as one of the first fellows in my independent laboratory. And Terry made this very important discovery that when our patients become profoundly lymphopenic, this is associated with a response. And that response is an elevation in available interleukin-7. And it was really the kind of data that was almost too good to be true. It was the mirror image of one another. Terry got to where he could predict, whether a patient was lymphopenic by looking at the ELISA plate. And so you felt really confident that you'd uncovered something fundamental. Interestingly, we made this observation in people long before we were able to make it in mice. Because of the technologies were just not there in mice to be able to measure it. Subsequently, we were able to demonstrate using the mouse model that the reason the IL-7 rises is not that the body produces more, it's simply that the body utilizes less. And several other groups subsequently confirmed our findings, and this really became sort of a new paradigm, the elevation in homeostatic cytokines that is responsible now for the change in homeostasis, the change in T-cell physiology that occurs with lymphodepletion. We then took this a bit further, and this is in collaboration with Ron Gress's group by undertaking first-in-human studies with interleukin-7. And the agent behaved quite beautifully, I must say. It was not toxic, and it provided, in and of itself, a single maneuver, a dramatic augmentation in circulating and we believe even total body T-cell numbers. It essentially invoked homeostatic peripheral expansion. You can see here the elevation in CD4 and CD8 cells induced by IL-7, the fact that this is largely due to peripheral cycling and not thymic activity. And we were even able to image this using FDG PET imaging, which of course demonstrates increased metabolic activity of populations, and when you gave IL-7, you saw that activity in the lymphoid tissues, really quite remarkably, demonstrating, again, this wasn't just some change in the bloodstream. And so at the end of these efforts, we really had contributed a new paradigm to thinking about T-cell homeostasis. And that is that when an individual is rendered lymphopenic by whatever maneuver, this is accompanied by profound changes in nearly every aspect of T-cell biology. And the bottom line is that T-cells become much more reactive to almost any antigen that they are presented with. In some cases, for high-affinity antigen, you end up with just a larger response, let's say, to a vaccine. In some cases, to low-affinity antigen, you actually change the nature of the antigens that you're capable of responding to. Antigens that you would have normally ignored now become sufficient to induce antigen expansion. And then there's also the memory pool, which is largely TCR independent. And so about this time, again, I was lucky enough to be recruited back to the pediatric, now called pediatric oncology branch, that was being led now by Lee Hellman. And Lee was very frustrated with the problem of stalled progress for solid tumors of childhood, as we all were, and we continue to be. This is kind of the epidemiologic data from ending in 2006. Things haven't changed very much in the last 10 years, showing the progressive decline in mortality rate for both solid tumors and, this is leukemia, lymphoma, this is solid tumors. The point is that the rate of decline has been greater for hematologic malignancies. We still have a ways to go, but we really have made not so much progress against solid tumors, and there was even some really flattening of the curve for at least a decade in the 90s. And so Lee, well, this is an example of the kind of data. This was generated here in our clinical trials that the pediatric oncology branch had been involved in for years. And frankly, using standard therapy today, it doesn't look much different. If you have a patient with a sarcoma of childhood who presents with non-metastatic disease, this is their survival, not so good. And if they present with metastatic disease, it's just dismal. And so the immunology section was established to ask the question, can we improve outcomes with immunotherapy? And of course, we went back to what we knew. We knew that this lymphopenia was likely to be a problem that we were inducing in all of those patients. This is an example of a patient who comes to you with a new diagnosis of Ewing sarcoma. Here's their CBC. You can see a normal lymphocyte count when we get them. But when we're done giving them the treatment, now 8 months later or 6 months later, pretty profound lymphopenia. And there's a variety of publications out there that demonstrate relationships between outcome and lymphopenia. These are correlations. They aren't causations. But it raises the question that maybe just maybe, lymphopenia is diminishing our patient's capacity to eradicate, let's say, residual neoplastic disease. And so as an immunotherapist, I wanted to try to develop ways to take what we've learned about homeostasis and apply it to the problem of pediatric cancer. And at the time, all we really had available was to undertake approaches that could augment existing immunity. Okay? And so these are the kind of immunotherapies that you've heard about for many decades. This is, let's give a patient a tumor vaccine. More recently, let's use checkpoint inhibitors, which relieve suppression on T cells. Let's take tumor-infiltrating lymphocytes that are being induced by cause of the natural response and expand them and administer them. In order for this type of an approach to work, you have to have a tumor that has an antigenic target. The tumor has to express MHC. The immune system of the patient has to be intact, and you're likely to have to deal with this suppression that, of course, many tumors have, especially solid tumors. We went about this by going after the period of minimal residual disease that can often be induced even in the worst childhood solid tumors. This is an example of a typical patient that you'll see, again, play out in our clinic even today. This is a CAT scan through the pelvis, a large tumor, administered dose-intensive chemotherapy. You see the tumor burden goes down dramatically after chemotherapy and radiation. You've lost logs of tumor. But invariably, again, this patient had a small pulmonary nodule, the tumor is going to recur. And at this point, the horse is out of the barn. It's very hard to get back in control. But we reasoned that there's a window of opportunity here at the period of low tumor burden where we might be able to use immunotherapy. And this had happened to be the time when the patients were profoundly lymphopenic. So we developed an approach we thought might be able to enhance immune reconstitution and direct T-cell responses, this homeostatic peripheral expansion. It was pretty elementary at the time. It was a first-generation trial. Why it was important and why we had these notable publications were not so much because the clinical outcomes were good, but because we did important and intense biologic endpoints. And we learned that just giving autologous lymphocytes doesn't immune reconstitute your patient, that the dendritic cells that we were using really were not up to snuff, and that we had a problem of expansion of regulatory T cells, which was made much worse by administering IL-2. And so subsequently now, 10 years later, we started a second-generation trial, which was more complex, more involved. I would maybe even call it Herculean, the kind of trial that you can only do, I think, at the NIH Clinical Center. But we were asking fundamental questions and basically targeting the same population with a new diagnosis of sarcoma or recurrent sarcoma, asking them to come to the NIH for a tumor biopsy and for an autologous lymphocyte harvest. Then they go home and get their chemotherapy rendered into NED state or load burden state and come back for the immunotherapy. And the immunotherapy, again, very complex. And we used all kinds of cocktails to make a better dendritic cell. We depleted regulatory cells. I'm showing you this to show how hard we tried. And in fact, we did a good job in terms of immune reconstitution. The IL-7 was quite effective at reconstituting these patients' immune systems compared to patients who didn't get IL-7. And patients with newly diagnosed metastatic sarcoma actually have remarkably good overall survival and were treated on this trial. So that's very encouraging. The problem is I don't know why. I don't know what the tumor antigens might have been in my tumor lysate. I know that those that had a response to the tumor lysate are doing better. But it really is very hard to take this therapy forward when it's so complex and you really don't know what is your mediator of response. Furthermore, when you measure the response to tumor lysate, when I say what's positive, let me tell you, we are talking a very, very low number of tumor-reactive T cells. Our maximum level was 0.014% in the best patient that we had. And I want you to remember that number because you'll see it later, or I'll refer to it later. And of course, you know, during the 90s, what these tumor antigens might be, a lot of individuals, including Steve Rosenberg's group, had done work on this. But it became clear that it could be almost anything. And so we really had a difficult time trying to pin this down. And in fact, more recently, there's been sort of a groundswell. I don't know whether this is going to eventually end up being dogma or not, but many people believe now that the immunogenicity of a tumor relates to its the degree to which it is mutated. And the more mutations a tumor has, the more likely it is to be immunogenic, and there's plenty of evidence that these mutated genes can be antigenic. And this is driven in part by the fact that melanoma happens to be one of the most mutated tumors out there and also one of the most immunogenic. But look what's over here. If you just look along the span of cancer, What are the tumors that are the least mutated? It's our pediatric solid tumors. And so it sort of raises the prospect that maybe our children are not going to be able to generate the kind of immune responses that an older patient with a more mutated tumor would be. Melinda Merchant in our group, who's led many of the clinical trials that I'll be discussing today, led a trial of the first checkpoint inhibitor in pediatric cancer. This is another way of getting at this question: can we uncover some kind of an existing immune response? And of course, ipilimumab, this is an antibody that blocks signaling via CTLA-4. So if you have a T cell that can recognize an antigen, by treating with ipilimumab, you might be able to turn that T cell on. 30 patients were enrolled on this trial, 30 children. We knew that the drug did what it was supposed to do because the children got plenty of autoimmune side effects, some of them quite severe. But despite this, we had no objective responses. 0 out of 11 children with melanoma, and 0 out of 19 non-melanoma. There was a little bit of a hint of increased survival if you had an immune-related adverse event, but this is hardly anything to get excited about. And so we sort of went back to the drawing board and said to ourselves, augmenting existing immunity may not be good enough. For whatever reason, the natural immune responses that our patients have to cancer may not be of high enough potency, and this could be that pediatric tumors have lower immunogenicity. And so another way to think about this, and this is where, from my point of view, the field has gotten very, very exciting, is that we can now start to sort of turn this paradigm on its head and say, wait a minute, maybe we don't have to rely upon natural immunity or upon naturally immunogenic antigens. Maybe we can train the immune system to recognize a molecule as an antigen that it wouldn't otherwise do so. And this is really moving yourself into the realm of synthetic biology. And of course, monoclonal antibodies are a perfect example of synthetic biology having an impact on treatment of cancer. We've been focusing our work, as Michael alluded to earlier, on genetic engineering of T-cell populations, in particular, chimeric antigen receptors. And so for the rest of the talk, I'm going to talk about some of our efforts in this area. For those of you who haven't followed the field, so of course a T-cell receptor recognizes a peptide in the context of MHC. It requires signal 1, but also, especially in the case of naive T-cells, it requires a second signal, and the prototype would be CD28 to get fully activated. And on the other hand, a chimeric antigen receptor that is currently being used essentially by all groups now in 2015 has already incorporated the second signal. And so that once the antigen is recognized, you get signal 1 and signal 2. And the other difference with a chimeric antigen receptor is that it is not reliant on MHC and on peptide processing, but rather utilizes usually a fragment of an antibody, a single chain FV, to recognize a cell surface molecule. So it's non-MHC restricted. And again, you get signal 1 and signal 2 when it fires. We're working on several CARs in the lab. I'm going to talk briefly about 19, targeting CD19, CD22, and GD2, and 2 different co-stimulatory domains. I want to give credit where credit is due. This is an area that's gotten a lot of press. Zeli Geshar is the first person to come up with the idea for a CAR. He actually was on sabbatical here in the surgery branch. When he did this work, it was a really out-of-the-box idea, and he wanted to somehow get T cells out from under the MHC restriction problem. This was 1989, and it took, as you can see, essentially 20 years to take this idea and move it to something that had any kind of activity in the clinic. This was investigator-initiated work in academia. This was not done by industry. And frankly, it wasn't all that well thought of for the first about 18 years. So this really shows that sometimes plugging away pays off. And the first payoff really came from this institution in 2010. In this publication in Blood was the first report of the activity of a CD19 CAR against in this case it was a lymphoma. This CAR was generated by Jim Kokendorfer in Steve Rosenberg's group and targeted CD19 and used a CD28 co-stimulatory domain. So we saw very early of the potential for using this in childhood tumors, and so we initiated a study using this same CAR, a little bit different way of making the cells, and targeting B-ALL in children. This trial is led by Daniel Trey Lee, an assistant clinical investigator in our branch. And it sounds difficult to do this. It sounds like this must be so tedious and involved, but I will tell you that from my point of view, I've been most impressed by how feasible it is to generate these products. Now, we have an outstanding Department of Transfusion Medicine in the NIH Clinical Center. These are very sophisticated folks who have been doing cell therapy for some time. But I still think that the notion that this is just too difficult is indeed rapidly fading away. And so the way we make these cells is we do apheresis the patient. We do an enrichment step with CD328 beads to kind of pull the T cells away, let's say from contaminating non-T cells. The cells are then exposed to a viral vector and undergo usually a second exposure, so 2 transductions, and then are expanded ex vivo. And then very, very importantly, back to our early days on the homeostasis issue, if you don't give chemotherapy to these patients, it really doesn't work. And there's been several groups that shown this, not necessarily in randomized trials, but I think there really isn't much in the way of this field that have not bought this idea that a lymphopenic patient who's been treated with some kind of cytotoxic therapy is much more receptive to the expansion of these cells. And it's been one of the major points, I think, that has allowed this field to progress to where it is today. And so we published these results last fall in The Lancet. And importantly, these are the only intent-to-treat results out there. That means everybody who came to the NIH and signed a consent form to go on this trial is reported in the report. These are not patients being apheresed on one trial and being selected or told to go home and wait 3 months and then come back and treated on another trial. And so I think it's really important to point out that this really is a proper intent-to-treat trial. All patients were treated with the same chemotherapy as well, and it was a very modest chemotherapy. I don't think anybody in the field of leukemia would think that our refractory leukemia patients would be rendered into remission with this regimen. 21 patients collected, all infused with CAR cells, median age 13 years, median blast percentage 28%. We had some with low burden disease and some with high burden disease. 6 of these patients had never been in remission despite multiple rounds of chemotherapy. And those patients, to my mind, are highly informative because the fundamental question you want to answer as an immunotherapist is your therapy doing something that chemotherapy can't do? Are you able to eradicate the chemo-resistant cells? And the results in these 6 primary refractory patients really were quite gratifying as they all were rendered completely free of disease. 90% of the patients were feasible. They were all infused, but 2 of them were not feasible because the numbers of cells generated didn't meet our bar, although one of those patients did have a good remission and remains alive and well. And the median transduction efficiency, you can see, was high. This is our waterfall plot for the 20 patients who had ALL. The patient who had lymphoma did not respond to the therapy. And this shows the change in marrow blasts in these patients, and you can see that everybody here in the light blue was rendered into a complete response that was completely negative by our most sensitive measures of monitoring, the flow cytometry, less than 0.03%. 2 patients were rendered into a CR, but were sort of flow cytometry positive. And for some patients, this was accomplished with minimal toxicity, grade 1 or 2 cytokine release. And for others, there was more toxicity. And I don't have time to talk about cytokine release syndrome today, but I'd certainly be happy to do so with anybody who wants to in the future offline. But so it was remarkable. Remember I showed you the 0.014% and everything we had to do to get to that number. And this is a patient who, is probably one of the highest levels, but it's not necessarily out of the ordinary. These are the CAR cells, looking at the CAR cells with an anti-idiotype on day 10. 79% of this patient's T cells are tumor reactive. We are talking orders of magnitude beyond anything we can accomplish with these other approaches that are simply vaccines or checkpoint inhibition. And coincident with the expansion of these cells, we see eradication of CD19 positive cells, both the malignant ones and the non-malignant ones. Now, interestingly, although the expansion was dramatic, so was the contraction. These cells disappeared. And this has been bothering us, and we're scratching our heads, and I'm going to talk a bit more about that in the last half here. Importantly, these cells do migrate to the central cerebrospinal fluid. They appear to be able to eradicate tumor in there. In 2 patients who had measurable disease going in, the CAR cells go in and the disease disappears. For patients who are rendered into an MRD negative remission, the outcomes have been good, although most of our patients go on to receive transplant. because we just don't know whether the remissions are going to be durable. And that is considered standard of care for patients with refractory leukemia who are rendered into remission. So to summarize, with this single infusion of this one cell product, small cell dose, we've got a 70% complete response rate in refractory B-cell ALL. This is an intent-to-treat analysis, no pre-selection. We've seen complete responses in all 6 patients who had primary refractory ALL. So it really confirms for me that this is doing something that chemotherapy just can't do. And I think the data is very clear that this is an effective bridge to transplant in this population. Whether this is curative in its own right is not entirely clear. They traffic to the tissues and mediate anti-tumor effects. We had a 14% grade 4 cytokine release syndrome. We were able to treat those successfully. It's an important area that I just don't have time to talk about today. But we observed B-cell recovery in all patients. And again, back to this question. Why didn't the CD19 CAR cells persist? Because people that have been involved in the adoptive immunotherapy of cancer have been sort of stating the mantra for the last decade or so, is that persistence is required. Now, persistence can be defined differently in different trials. Sometimes it's persistence at a month. Sometimes it might be persistence several years. But I think if you want to eradicate especially large, bulky tumors or tumors that are living in a small little niche somewhere, I think we all believe that we can do better if our cells persist. And so one possibility is that there is an anti-CAR immune response. The single-chain Fv in this case is a murine, and there's no reason that it shouldn't be immunogenic. And in fact, we looked for human anti-mouse antibodies. We didn't find them, although the single-chain Fv is not usually the area where HAMA are directed. We looked at T-cell proliferation in response to CAR transduced versus mock transduced cells in 11 patients from whom we had adequate sample. And we did find some T-cell responses to the CAR. In general, the proliferation index to CAR transduced cells was higher than that to non-transduced cells. And in some patients, that actually preceded the administration of the CAR. But we found it more often in patients after administration of the CAR. But even in patients who had the responses, we often had a good response rate. So we couldn't correlate this immune response with the immune response to the CAR, the T-cell proliferative response to the CAR, with the eradication of the leukemia. And so we conclude that T-cell mediated anti-19 CAR responses are commonly found, that they don't preclude a response to therapy, and they might contribute to diminished persistence. And certainly, I think the field is moving towards thinking about using fully human single-chain Fvs, and whether that should diminish this reactivity, it may not take it away completely. But as Michael mentioned, I'm leading this multi-institution consortium that has really brought in investigators from around North America who are engaged in this type of therapy. And the goal of the grant is not only to identify new targets using genomics for solid tumors, but also to increase collaboration amongst those of us who are conducting CAR therapies so we can learn more about how best to optimize these and iteratively improve these. And in particular, this represents the Penn group, the NCI group, and the Seattle group. And we each had a very similar CD19 CAR in the clinic, using the same single-chain Fv, a similar expansion platform. And the notable thing is, whereas Penn noted prolonged B-cell aplasia, as have the Seattle group that uses 4-1BB, our CD19 CAR is not associated with prolonged B-cell aplasia. So we see the 19 cells go in, the CAR cells go in, expand, and then disappear. And so it was the first kind of clue that maybe the co-stimulatory domain was playing a role in impacting persistence of these cells. And of course, CD28, something we all know, we think we know a lot about, member of the immunoglobulin superfamily. As I mentioned, the prototypic signal 2 for naive T cells signals through a variety of pathways. The counterpoint in the cells, the CARs that were inducing prolonged B-cell aplasia was 4-1BB. It's a completely different family, a TNF receptor superfamily, but signals through similar pathways. And so we really didn't have a good feeling for why these should be necessarily different. But we began to hypothesize that maybe the difference in the persistence relates to the difference in the incidence of T-cell exhaustion. And T-cell exhaustion is something that immunologists have been working very diligently on, both mouse and human, for the last several years. T-cell exhaustion is something that happens in disease states. So under normal circumstances, a T-cell is presented with an antigen. The T-cell responds, the antigen disappears, the T-cell becomes a memory cell, a polyfunctional memory cell that can make a lot of cytokines and proliferate. But if the antigen stays around and keeps signaling the T-cell receptor, what you see is a progressive loss in functionality of the T-cells. And this is associated with diminished cytokine production, diminished proliferative capacity, and increase in the expression of checkpoint receptors. Of course, checkpoint receptors are those receptors that are now being targeted with things like anti-PD-1. These are inhibitory signaling receptors that are presumably there to prevent overproliferation. So I'm going to change gears now and share some data that we generated using a completely different CAR that ultimately came back and shed light on, we think, why we observed this short persistence in our CD19. So GD2 is a ganglioside. It had already been vetted as a tumor antigen, at least for monoclonal antibodies, in pediatric neuroblastoma. And we had looked at it in pediatric sarcomas, and you can see here how high GD2 expression is in osteosarcoma and some of our rhabdomyosarcomas. And so we reasoned that, again, getting back to this goal of trying to target pediatric solid tumors, if we could make a CAR targeting GD2, perhaps we could improve outcomes. And so we did this in the laboratory, and this is work that's really been led by an incredible MD PhD student in my lab, Adrienne Long. And what Adrienne observed was that, the GD2 CAR, and this had a 28 co-stimulatory domain, functioned pretty well in the test tube against osteosarcoma cells. But when you put it into mice, it really didn't do much of anything against the osteosarcoma. And this was in contrast to the CD19 CAR, which also, had reasonable activity in vitro in terms of killing. But using a bioluminescence model here, you can see how well you were able to eradicate leukemia in these mice. Well, we saw this data and we said, yeah, but that's leukemia. We're trying to cure solid tumors here. Solid tumors are a lot more difficult. This probably isn't the fact that the CAR is any different, it's rather that the tumor is different. And so Adrienne rose to the occasion and said she was going to get to the bottom of this disparity between good in vitro killing and poor in vivo efficacy. And so she created an osteosarcoma cell line that expressed both GD2 and CD19. And we asked at that point, how good is the CD19 CAR? And we fully expected to find that the solid tumor was just more difficult to kill. But in fact, we were surprised. And in fact, even though the osteosarcoma was equivalently killed by the 19 and the GD2 CAR in the test tube, only the 19 CAR was able to shrink the established osteosarcoma. So it wasn't that solid tumors are impenetrable or you can't target them with these types of therapies. It was that there was something wrong with our CAR. And at this point, we went back to the drawing board and said, well, what's the difference between the two CARs? The big difference is the single chain FV, the co-stimulatory domain. The rest of the signaling domains are the same. And Adrienne began to study these really prior to the time that they went into the mice. And so you take a donor's T cells, you activate it with CD328 beads, a little bit of IL-2, you add your vector, and this is now before you even challenge this with antigen. And when she looked on day 7, these GD2 CARs already started to look different. They showed signs of what we call overactivation. Whereas the mock transduced cells that had the same CD328 beads and the CD19 CAR expressing cells had already settled back down by day 7 and their activation marker expression tended to normalize, these GD2 CAR cells remained activated as evidenced by larger size, high CD25, high 4-1BB, low IL-7 receptor, low 27. It was exactly what you'd expect of a cell that was continuing to get antigen. And in fact, when you look at the zeta phosphorylation, you can actually distinguish the zeta associated with the CAR from the native CD3 zeta. And you look at phosphorylation of zeta. In fact, here was the smoking gun. You had the GD2 CAR already phosphorylated zeta without any antigen in the system. And this was very different than the 19 CAR, which was pristine. And then that followed by the acquisition of these checkpoints. And you can define exhaustion any one of a number of ways, but PD-1 expression is a good place to start, and this was very high on the GD2 CAR. TIM3 and LAG3 very high on the GD2 CAR. We certainly wanted to rule out the presence of antigen, and I don't have time to go through all of this data. Tonic signaling. There you go. So we did rule out that there was antigen in the system. GD2 is not present in the culture system. Adrian made point mutations in the CDR3 region that ablated the capacity of the GD2 CAR to recognize antigen, and yet the exhaustion phenotype remained. Fusion protein incorporating the binding domain did not bind to any of the cells in the system, ruling out cross-reactive antigen. And so we ended up with this working model that this CAR was signaling without antigen. And how could that happen? And so you start to think about, well, what's on the surface of this cell? Well, it's a bunch of single-chain FVs, which notoriously oligomerize, certainly ex vivo. The antibody community has told us this. And it's usually because of interactions in the framework region. And so we began to reason that maybe there was spontaneous dimerization happening. And so an undergraduate in the laboratory, Kelsey Wenhanen, tagged our CAR with a fluorescent protein. And indeed, we were able to actually visualize this, I think, quite remarkably, better than we ever even expected. The CD19 CAR, and we estimate there's about 1 million of these on the surface of a T cell. We're talking really high level of these proteins. But the CD19 CAR distributes beautifully, homogeneously around the surface of the cell, whereas the GD2 CAR is consistently punctate. It looks like it's aggregated, and indeed, if you look at fret, it's clearly fretting with itself. We tried to localize the problem, and we were told that it was likely to be the framework regions. And so we began to do kind of a series of swapping experiments where we said, well, the CDR3 region, these are the CDR regions, 1, 2, and 3 are the antigen binding domains. The framework regions shouldn't be antigen binding. So if it's the framework region of the GD2 CAR, if we put that into the CD19 CAR, we should reproduce the phenotype, and vice versa, we should fix the CAR. Well, we were able to reproduce the phenotype. When we inserted the framework region from the GD2 CAR into the CD19 CAR, what we saw was the presence of exhaustion. This is indicated here by the high PD-1. Unfortunately, we were not able to fix the CAR because when we put the other, we were not able to get expression of the CAR on the surface. And that's been difficult for us to do, to localize the area on the single-chain FV and specifically mutate the area that's aggregating. It's likely to involve multiple amino acids. But importantly, we don't think this is unique to the GD2 CAR, because when we've looked across a variety of CARs, the GD2 CAR is the most obvious. But looking at a variety of CARs, a HER2 new CAR, a CD22 CAR, 2 different CD22 CARs, we see this phenotype of early exhaustion and the whole shebang. And showed here is the sort of pie charts where you can just see that MOC and CD19 are the most pristine. And then as you move over here, you get more and more tonic signaling until you have the GD2 CAR, which is full-blown exhausted. So to summarize, early CAR T-cell exhaustion is good killing ex vivo, but poor persistence, poor activity in vivo. CAR T-cell exhaustion is associated with tonic antigen-independent signaling that results from aggregation of the single-chain FVs. Most CARs show some degree of this phenomena. GD2 is the worst we've seen, but CD19 is the most pristine, raising the notion that maybe that's why the CD19 is so good. Now, this also opens up a possibility, because here now the scientist in you comes out and says, look, we have exhaustion in a dish. Immunologists have been wanting to study exhaustion for years, but normally you can only find an exhausted T cell in a patient who's had a chronic infection. Here we can start with homogeneous cell populations, and in 7 to 9 days, you've induced an exhausted T cell. We begin to ask, what is the biology that surrounds T cell exhaustion? And this thing never comes out okay. But basically, what Adrian was able to do is to start mutating various aspects of this synthetic receptor, either the zeta chain or the CD28 chain, to see what is necessary to induce exhaustion. And the bottom line is that CD3 zeta is necessary and sufficient for the exhaustion, but CD28 makes it worse. So CD28 signaling along with zeta makes a cell more likely to become exhausted. And you can look at this using transcription factors or using your checkpoints. On the other hand, 4-1BB is protective. So 4-1BB co-stimulation leads to much less expression of these checkpoint molecules, much better cytokine production when the cell is challenged with antigen, and improved anti-tumor activity in vivo. So finally, we're able to get an anti-tumor effect using the GD2 CAR against this osteosarcoma, simply by changing the co-stimulatory domain and preventing exhaustion of these GD2 CAR T cells. Now you say, okay, well that's unique to the GD2 CAR. It might be the only CAR that acts like this. How important is it to other CARs? Well, in fact, when we go back and look at the CD19 CAR, because you don't have to get exhausted by tonic signaling, you can get exhausted by high antigen burdens, we see that indeed the 4-1BB co-stimulation leads to more persistence, and these are in mouse models now, in the blood, similar to what was seen in the humans, and diminished expression of these checkpoint molecules. And furthermore, in a third CAR, this is a CD22 CAR that was generated in my laboratory and now is being in the clinic targeting patients with CD19 negative leukemia. Again, this is work that was generated by Terry Fry, that indeed the 4-1BB version of the CD22 CAR is also more effective and more persistent. And in fact, that's the trial that is open now in our branch and being led by Terry Fry. So again, it's exciting to make a better mousetrap. It's exciting to make a new therapeutic that works. But it also provides an opportunity to learn something about basic biology. And so we thought we now had this cell in a dish where we had either very similarly treated, except very exhausted, and this sort of rescued exhaustion. And so we began to map gene expression in these different populations. And we saw that, indeed, they localized with the 28 zeta CAR. The exhausted was very different than what you found in the mock and the CD19 28. And that the BB modified the gene expression, but not back to these. And so there was a unique subset of genes that the BB provided that modulated exhaustion that gave us a clue about the biology. And some of these were the usual candidates. So exhaustion-associated genes were quantitatively diminished by BB, and memory-associated genes were quantitatively increased. And that was gratifying, but perhaps not surprising. What was a bit more surprising is when we used an unbiased analysis of this gene expression profile. So don't go look for something that you already know has been reported in exhaustion, but just say, what is the most important distinction between the BB expressing rescued and the 28 expressing exhausted? And we began to find gene families that are not what you'd expect. Response to hypoxia, cellular metabolism. Apoptosis didn't really surprise me. But really, response to hypoxia as a mediator of exhaustion is not something that's been reported before. And so I think this opens up new opportunities to study the biology of exhaustion. In fact, the metabolism one is one that we've been quite interested in, mostly because early on we noticed that these cells look different. I told you that even by day 7, when they were highly activated before there was any antigen, what Adrian had noted is that they always acidified their media. The CD19 CAR in the mock cells, the media stayed pink. This wasn't because there was more cells in the media. There were actually less cells there. They just had a higher glycolytic rate, a bit of a higher OXPHOS, but a higher glycolytic rate, and a significantly higher mitochondrial mass. So it was clear that these cells were metabolically deregulated, and whether this was simply a symptom or causal of exhaustion is an area, I think, of really important question to answer. We've gotten some interesting clues of late using approaches to modulate glycolysis. This is essentially Warburg that these T cells are doing. And so we're thinking of ways, similar to the cancer biologists, to modulate Warburg and to see whether it might repair some of the functionality of these cells. And I can tell you that diminishing the glycolytic rate, in this case by providing galactose, can substantially improve the cytokine production of this exhausted CAR. So simply modulating a metabolic pathway may be sufficient to increase the functionality of a T cell. And interestingly, that same effect is not seen on the 19 CAR, which is an exhaustion, suggesting that this is somehow an exhaustion-specific phenomenon. So the second summary is that CD28 signaling in T cells exacerbates exhaustion. And in contrast, 4-1BB signaling induces an anti-exhaustion program in chronically activated T cells. And that these features likely explain differential persistence that has been reported for 19BB zeta CARs compared to 1928 CARs. We propose that persistent CARs are likely to be more effective against cancer. And so we think that going forward, we will probably focus most of our efforts on BB CARs. Despite the vast amount of work being done in human immunology, I think we still have a pretty superficial understanding of T cell exhaustion. And this data has provided us with novel insights that metabolic changes and hypoxia response pathways may be important. And just to conclude, so I hope I've demonstrated to you that the bench to bedside to bench approach is an efficient one for illuminating new biology and for developing new therapies. And I think there's no better place to do this type of work than in the NIH Intramural Research Program with its laboratories and clinics. Technological advances in synthetic biology are yielding novel therapeutics crafted in academia. You can make these things in your lab, which is remarkable. But it's not just that they're new therapeutics. I think these are also providing novel and elegant models for the study of biology. And I hope I've convinced you that we've come a long way since I read that article in 1985. It's really an exciting time for the immunotherapy of cancer. I want to thank the people who have contributed to this, the clinical team in the pediatric oncology branch. We have an incredibly dedicated clinical team. I've mentioned Melinda Merchant, who really leads the immunotherapy trials in solid tumors, Trey Lee, who led the CD19 CAR trial, Terry Fry, who is our leukemia expert and is leading the CD22 trial, Nurali Shah, who provides invaluable support for those patients, and our research nurses, Donna, Amanda, Cindy, and Bonnie, who really make this stuff happen. Sharon Mavrakakis is our regulatory expert, and Joanne is our nurse practitioner. I've highlighted work from my section. I mentioned Adrian's work. Wah has done a lot of the work on monitoring of our patients. Karen Ling, Alec, Jillian. Jillian's done some of the recent work on metabolism in the lab, and Waleed was responsible for the CD22 work. I couldn't have done these trials without the incredible support of the NIH Clinical Center, especially the Department of Transfusion Medicine. These people absolutely work tirelessly to churn out these products with relatively limited resources. This program is led by Dave Stronchek, Joe Proctor. I really am thoroughly and completely indebted to them. As I mentioned, Jim Kokendorfer made the CD19 CAR. Steve Rosenberg graciously has provided the vector that we have used in our trials, and Mary Alice provides important monitoring for our patients. So I'll stop there and take questions. Thank you.
Yes. In pediatric pathology, it's well known that a neuroblastoma can spontaneously regress. How does that relate to any of this? And wouldn't that shine light on how to best attack what we used to call the small blue cell group of tumors in general?
Yeah. You know, that provocative observation has plagued us. Many investigators hypothesized that was immune-mediated. I believe that most investigators now feel that that's a differentiation program. When they regress, what they usually do is differentiate. There's also some differentiation to more mature elements. Retinoids and epigenetic changes can cause these cells to differentiate. So I can't assure you that it's not immune-mediated, but I would suggest that the prevailing thinking in the field is that this is more of a cell-intrinsic differentiation and not an immune reaction.
Thank
you.
Crystal, two things. Do you know if the BB versus the CD28 is changing the pattern of expression on the cell surface?
No, it absolutely is not. In fact, the tonic activation is the same, and the activation expression is the same. And the Warburg is still the same. They still make the media turn yellow, but they're just not as exhausted. So it's providing some kind of protection against the exhaustion.
And the second thing is, if you want to work on myeloid malignancies and have targets that are also expressed on stem and progenitor cells, your situation of a hit and run is actually kind of perfect.
You're absolutely
I
right.
mean, the people that are trying to develop CARs against CD33 and IL-3 receptor alpha and all, are really worrisome in terms of, even a few of these T cells, you think they stick around, you're going to be pancytopenic for life, whether it's allo or auto. Is there a way you can actually predict this enough to...
Well, I don't know, Cindy.
Like in my
It's
monkeys,
still
to
new.
do this first, right?
Yeah. You're going to incorporate a lot of protections in there. You're going to incorporate a suicide vector and all kinds of things.
Yeah, which are never 100%, so that's,
Yeah, no, but I agree. The hit and run of the 28 could be very useful for like an AML CAR. Absolutely.
Brilliant data. So a lot of these patients with heme malignancies come here having received lymphodepleting chemotherapy to try to get rid of their leukemia, lymphoma. And so the apheresis product, in effect, is growing up in this lymphopenic environment. So have you looked at all the extent to which just the pheresis product or the T cells in the pheresis product are already coming out perhaps more differentiated or exhausted than in a patient who hasn't gotten prior chemo?
Yeah, that's a really good question. And I gotta believe that's playing out here somehow. It's actually remarkable to me that we're even able to grow products from most of these patients. And the fact that they're able to go into their patient and expand, I'm flabbergasted because, as I showed you, I spent the first years of my life showing how messed up their immune system is, and yet you just put a gene in there and this thing becomes an army. So I think that there probably is a situation where, you're beyond the capacity to be able to rescue those cells, but it's surprising how effective they've been.
And a second question, if I may, I hate to monopolize. How would one sort of decide whether or not CAR therapy is a destination rather than a bridge to transplant? What kind of trial would have to be designed to have that as an endpoint?
Oh, that's a really good question. I mean, my own feeling is it's going to be hard to answer that in children. Certainly, we're going to learn it in the multiply transplanted population first, because each subsequent transplant is less effective. So if we can demonstrate, it's already questionable whether we should be transplanting patients a second time that are put into remission. And certainly a third transplant, come on, it's the toxicity is high. So I think we'll learn from those patients. We'll probably also learn from the young adult population where the current way of treating young adults with ALL really isn't that great. And the toxicities are, I think, less tolerable. So it's going to take a while to come around to that and figure out where does this thing fit in. It's a big challenge. It's probably going to be done by the clinical researchers rather than, the people that run large trials rather than people like me. But.
Thank you for this wonderful contribution that you've made to pediatric oncology, first of all, and your wonderful talk. I just was wondering if you had any idea how the patients with refractory ALL did post-transplant. And then secondly, in terms of the T-cell exhaustion, whether that has anything at all to do with thymic involution.
Yeah. So the post-transplant course of these patients, we believe to be, again, the numbers are small. But we haven't seen anything to make us think it's different than the usual post-transplant course. One patient, unfortunately, died of graft versus host disease. Again, that's the kind of thing that happens. This is a second transplant for him. So I don't think it's changed the outcomes post-transplant with all the good and bad of that. As far as the exhaustion, this kind of gets to what we were talking about, that the more these cells have been driven, the thymic-drive cells are new again. They're the antithesis of exhaustion. And so whenever we're relying on cells that haven't been through the thymus in the recent past, and I'm talking number of divisions, you're going to be more likely to be exhausted. So if you come in already on the verge of exhaustion, then you stick that CAR in there and you're tonically signaling it, you're going to get to the point of no return more quickly. So I think it is all wrapped up in the same package, but those complexities we haven't been able to, this is just an idea. We haven't shown data to that.
Crystal, I have one additional question.
Yeah.
It related to the earlier question about using the CD19 CAR for primary treatment of leukemia in children. One of the points that you made is that these are chemo-resistant patients, so they've had multiple rounds of chemotherapy.
Yep.
Given what you know about homeostasis and lymphocytes, is that contributing to their response? Is that a necessary component, or do you think that's unrelated?
No, it's interesting. It could. It could. I don't know. The primary refractories, I guess, are reassuring because those are had often only been diagnosed, but they'd had multiple rounds of chemo, but they hadn't had years of chemo. I don't know, Michael. It's a good question.
Yeah. And of course, we're interested in drug resistance and that cells themselves are changing because they're resistant for a reason. They have physiological changes that may also be contributing to their sensitivity.
Yeah. They may be less fit.
Right.
Right. Absolutely. Yeah.
All right. If there are no further questions, let's thank Dr. Mackall for an amazing talk. There will be a reception in the library, and you can ask more questions there.
Thank you.